In the surgical department of a hospital, there was an outbreak of hospital infection that showed itself in often postoperative wound abscesses. Postoperative wound abscesses. Biological examination of a pus revealed aurococcus. What examination shall be conducted to find out the source of the causative agent among the department personnel? What tests should be done? Don't forget, this is what postoperative wound abscess. Postoperative wound abscess. And over here, you want to determine the strain of bacteria, or you want to know the strain of bacteria, whether it is gram positive, gram negative. You want to know the strain of that bacteria. Is it spore? Does it give spores? Does it ferment? Does it undergo coagulation? Or does it coagulate with milk? You want to know the strain, the single strain of the bacteria. And this method is called phage typing or phagotyping. It is called phage typing or phagotyping. And it is used to trace the source of outbreak of infection. It is used to trace the source of outbreaks of infection. Outbreaks of infection. So here you are looking at what? Phagotyping or phage typing. Phage typing. Again, you are looking at what? Outbreak of an infection. Outbreak.
Please excuse me, I couldn't hear that word well.
Phage typing. Phage. P-H.
Phage is used to what?
I'm saying it is used to trace the source of outbreaks of infection. The source used to trace the source of outbreaks of disease. Phage typing. Phage typing. Phage typing. To trace the source of outbreak of a disease. And over here, we are in an outbreak hospital. Outbreak hospital. Outbreak hospital. So again, you are thinking of phagotyping. Phagotyping. So your answer is to be E, not C. Your answer is E, not C. It's E. It's E, not C. All right. It's E, not C, please. A 7-year-old child suffers from streptococci angina. Doctor suspected development of rheumatism and administered surgical examination. First of all, if you have strep and you are having what? Rheumatism, how do we call it? We call it what? Rheumatic what? Fever. And because it's rheumatic fever, it means that you're having a strep what? Infection. Streptococcal what? Infection. Streptococcal what? Infection. So that's what group A. Group A. We have done these things before in many of the questions that we've done. Now, the provisional diagnosis was confirmed by the presence of antibodies to the following what? Antigen. So, how are you going to confirm the presence of strep infection? Strep infection. The answer is what? O streptolysin. O streptolysin. O streptolysin. Or anti-streptolysin O is the antibody that made against streptolysin O an immunologic reaction. Do you understand? So this is to the antigen, you are producing what? Anti-streptolysin what? O. Anti-streptolysin O. That means this O streptolysin is actually what? The antigen. So you are giving what? It's anti-streptolysin. Anti-streptolysin. So the answer is what? Is A. Is A. O streptolysin. All right. So we're having a culture of monkey cells. And a group of mouse sucklings were infected with a inoculum taken from a child with provisional diagnosis of what? Enterovirus infection. There was no cytopathic effect on the cell culture, but mouse sucklings died. What enteric viruses might have caused disease of this child? what might have caused the disease of this child. So first of all, you must understand that caustic is the causative agent for enterovirus. Is the causative agent for enterovirus. The causative agent for what? Enterovirus. We call it caustic virus. Caustic virus. And we have the A and the B. The A is more severe that it can lead to paralysis and death of the microorganism. In this case, the mice. Whilst COX-B causes less severe infection. It will cause a less severe infection. So since the mice died or the mouse died, we could be thinking of what as a severe case. And so therefore, we'll go for what? Caustic A. Caustic A. Caustic A or caustic virus A. All right. A patient has been suffering from elevated temperature, attacks of typical cough for 10 days. Cough for 10 good days. Doctor administered inoculation of mucus from the patient's nasopharynx on the alga. What microorganism is presumed? Look, cough, persistent cough. So you are thinking of what? Pertussis or Bordetella pertussis. You are thinking of Bordetella pertussis. Bordetella pertussis, also known as Pertussis bacillus. Pertussis bacillus is a gram-negative bacteria. Gram-negative bacteria. Okay. Guys, look at it. A patient of a surgical department complains about pain in the small of her back and at the lower part of her belly. Painful and frequent urination. Look, I think I've done this question already. Bacteriological examination revealed a gram-negative oxidase positive rod-like bacteria forming greenish colonies. Greenish. And I say if you see greenish, you are either thinking of what? Salmonella or what? Pseudomonas. So over here, your answer is what? Pseudomonas aeruginosa. Pseudomonas. So your answer is E. Is E. Again, if they say person having a red colony or it undergoes fermentation, then you are thinking of what? E. coli. You are thinking of what? E. coli. But this is not that. This is what? Pseudomonas aeruginosa. So your answer is E. A female patient underwent a transplantation. Liver transplant is type what? Of course, type 4, hypersensitivity reaction. Type 4, hypersensitivity reaction. And usually it is a reaction, we call it what? The delayed type or transplant rejection. They say 1.5 months after it had, her condition became worse because of rejection or transplant rejection. What immune system plays a role? Again, you are looking at what are the T cells. You are looking at what are the T cells. But now we are having T killers, natural killers, and so on and so forth. So actually the answer is, so this and this, they all mean the same. So I don't know why they brought the two of them to be present. Okay, so over here we're looking at what are the T killer cells or the T natural cells or natural killer cells. T natural killer cells. They play a major role in the host rejection. They play a major role in the host rejection. So your answer should be what? Should be A. But of course, it can also be natural killers, but the T killers will be more appropriate. But it's the same thing. It's the same thing. But in Krok, go in for the T killer cells. The T killer cells. Again, if we are thinking of immunodeficiency, what comes to mind? You are thinking of the T lymphocyte to be what? Helper cells. To be the helper cells. To be the helper cells. So some of these things, you need to know them. All right. So the answer is A. Microscopic examination of a microbial culture revealed fusiform spore-forming microorganisms that get violent blue gram stain. Violent blue gram stain. What microorganism are they talking about? This violent blue is specific for Clostridium or Clostridia. And it's a gram-positive, rod-shaped, anaerobic, spore-forming bacterium. Again, this is a gram-positive, rod-shaped, anaerobic. That means it doesn't like oxygen. So usually in tetanus and then in ulcers, you will see this Clostridium playing a big role. A big role. So we are thinking of what? Of Clostridium. And again, the staining is what? Violet blue. Violet blue. Violet blue. So your answer is A. Okay, a specimen obtained by the Oshensko method contains rod-like microorganisms stained with blue. It didn't say violet, it's what? Blue. Blue with round terminal components stained red. What are these components typical of? What are the components typical of? So write this down. Oshinsko method is for spore formation. Oshinsko is for spores formation. Whilst Burringens, Burri, Burringens is for capsules. It's for what? Capsules. Capsules. It's for capsules. Again, Oshinsko's method is spores. Burringen method is capsule. Please write it down. Please write that down. So your answer is what? Spore. Some of these you can explain it. It is as it is. It's a method. So I don't know, but it is what it is. During the regular sanitary epidemiological inspection of a pharmacy, the bacteriological analysis of air was performed. The air was found to have bacilli, yeast fungi, hemolytic strep, micrococci. Which of the following detected microorganisms indicate the direct epidemic danger? A direct epidemic danger. Direct epidemic danger. And over here, you are looking at what? At the streptococcus infection. Streptococcal infection. It is more what? Dangerous than the rest. So we're going for what? Strep infection. Strep infection. Strep infection. Because it can even lead to what? In surgical wounds, for instance, we have them all the time. Strep infection. So that can lead to what? Sepsis. So we're going for what? For hemolytic streptococcal. That one have or indicate a more direct epidemic danger. Epidemic danger. So your answer is D. All right. A bacteriological laboratory received a sputum sample of a patient suffering from tuberculosis. Bacterioscopic examination of smears and detection of the tuberculous bacillus can be realized by one of enrichment methods that involves processing of sputum only with solution of caustic soda. Only with solution of caustic soda. What is this method called? What is this method called? So first of all, the sputum for microscopic examination is subjected to three methods. Three methods. We have what we call the concentration method. We have the homogenization method, and we have the decontamination method. Decontamination method. So these are the three main methods that we use in microscopy or in sputum microscopy. We have concentration method, homogenization method, and then decontamination method. Decontamination method. Now, Petrov's method, P-E-T-R-O-F-F, Petrov, P-E-T-R-O-F-F. Petrov method uses what we call caustic soda to give good results. Petrov's method uses what we call caustic soda to give very good results. And homogenization may also be done by the use of these caustic sodas. By the use of these caustic sodas. So actually, we are referring to what? Homogenization. 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 So your answer should be what? Should be D. Homogenization. Homogenization. So why? Because it's involving the use of what? A caustic soda. That means it is borrowing the method of what? Petrov's method. It uses Petrov's method. It uses Petrov's method. Petrov's method. Petrov's method. So here you are thinking of what? Of D. D. Okay. A pregnant woman was registered in antenatal clinic and underwent complex examination of a number of infections. Look at this. The blood serum contained IgM. What is the result indicative of? IgM. IgM always indicates an acute or primary infection. It indicates acute or primary infection. It is also the first antibody to appear in response to exposure to an antigen. I remember when I was giving the immunoglobulins, the type of immunoglobulins, IgE, IgM, IgG, and what have you. I made mention that the first immunoglobulin to appear at the site of infection is IgM. 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 And again, it is an indication that there is what? An infection or primary infection. Primary infection. Now, in secondary infection or in reinfection, you will see an increase in what? IgG. You will see an increase in what? In IgG. 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 So how do I remember these IgG and IgM? IgM, most abundant. Okay? Most abundant. So if there's any infection, it will be the first people to go. Yes. So if you can remember that, then this is a primary infection because it's the most in the blood. Most. So that will go first. Secondary is IgG. So that is just by the way. So here we are thinking of what? A primary infection. Primary infection. What I just told you is just a clue to help you remember the IgM. All right. A 65-year-old man has purulent abscess on his neck. Again, abscess is there. Abscess. Analysis revealed gram-positive culture with, guys, look, plasmocoagulase activity. You remember when we were talking about the Staphylococcus aureus, the characteristics of Staphylococcus aureus? We said it is what? It has a positive plasmocoagulase activity. Positive plasmocoagulase what? Activity. And again, it is grape-like or just a grape-like clusters. Grape-like what? Clusters. Grape-like clusters. So please, all of these things help us to know that this is referring to what? Staphylococcus aureus. So your answer is what? Is E. Staphylococcus aureus. Staphylococcus aureus. Again, material taken from a patient with provisional diagnosis influenza was referred to the lab for virological examination. The hemoabsorption reaction was applied. Hemoabsorption. This reaction can be applied for detection of which of the following viruses. Guys, look, there is what? Hemoabsorption. Hemoabsorption. So definitely they are going to look out for what? For an antibody. 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 So this is what? Agglutination reaction. So they want to look out for what? Antibody. So what do? You're looking for what? For viruses containing hemagglutinins. And I said hemagglutinins refer to what? Antibodies. They refer to what? Antibodies. Antibodies. So your answer is what? It's A. It's A. It's A. All right. vomiting matters of a patient suspected of having cholera were detected in the laboratory. The material was used for preparing a hanging drop specimen. Hanging drop specimen. What type of microscopy will be applied for indication of the causative agent by its morbidity? By its morbidity. So first of all, you know that cholera is caused by what? Vibrio cholerae. Vibrio cholerae. And this is a gram-negative curved comma-shaped bacterium. And again, what does it grow? In which media does it grow? It grows in the alkaline what? Media. It grows in the alkaline what? Media. Now, how do you screen for the presence of this Vibrio cholera? We use what we call the dark field and then the phase contrast microscopy. Dark field and then phase contrast microscopy. These are used for screening of what? Fecal specimens for the presence of Vibrio cholera. The presence of Vibrio cholera. So in your option, you're either looking for what? For dark field or phase contrast microscopy. Dark field microscopy, phase contrast microscopy. These are used for detection of what? Of the enzyme. And again, which media will it grow? It will grow in what? In alkaline media. What is the shape? Curved, comma shaped. So please take note of these things. Inoculum from the pharynx of a patient ill with angina. Ill with angina was inoculated into a blood. Guys, do you remember I mentioned something about tellurate agar? I mentioned something about the tellurate agar. Let me see who was writing it. Who will I call? Who should I call? Lucy. Tellurate agar. What did I say it was for? I mentioned tellurate agar or Lufla. Lucy will not answer me. Yaira?
I'm here.
Good.
I'm here.
So tellurate agar. What did I say it was used for?
For
diphtheria.
Thank you. Diphtheria. Thank you. All of you are correct. Exactly. For diphtheria. That's why I tell you guys to write it down. If you have not written it down, maybe you could have forgotten it. So tellurate agar and then the Lofla is for what? It's for diphtheria. So detection of Corynebacterium diphtheria. So your answer is what? Even without going through all these things, your answer should be what? Should be diphtheria. I love that. It means you are listening. And I'm glad that you people are what? Are listening to the nonsense maybe I'm saying. I'm glad. And again, look at it. We say it resulted in a gray, radially striated what? Colonies. And in gray, we are referring to what? A fibrinous kind of what? Inflammation. And we said in fibrinous, we have what? Catarrh and we have what? Diphtheric. So you see? Great. So let's continue. This I won't waste my time on it. So the answer is what? Diphtheria. Again, look out for what? The tellurate agar and then the Loeffler. Loffler. Loffler. The Loeffler or the tellurate agar. Okay, so we'll move on. During examination of a patient, a dentist revealed a lot of white spot zones of enamel demineralization. White spot. What microorganisms take part in the development of this process? So what I'm saying is that what microorganism is involved in tooth decay? What microorganism is involved in tooth decay? And your answer is what? Streptococcus mutans. Again, what microorganism is involved in tooth decay? And your answer is what? Streptococcus mutans. And these are facultative anaerobic, gram-positive bacilli or coccus. Gram-positive coccus. So again, it's what? It's mutants. So the answer is what? Is A. Streptococcus mutans. Streptococcus mutans. All right. Okay. Planned mass vaccination. 5 to 7 days old children against tuberculosis. Tuberculosis. So what do you do? What vaccine is applied? We just said it in the last lecture. This is what? BCG. Or the Mantox test. The Mantox test. BCG or the Mantox test. The Mantox what test? All right. So the answer is what? Is A. Oh, guys, look at your question again. A 4-year-old child presents with general weakness, sore throat, deglutative problem. After his examination, a doctor suspected diphtheria and sent the material for lab investigation. Now, in order to determine the diphtheria causative agent, material should be inoculated in the following medium. Your answer is, thank you. No, it's A, not D, it's A. This is A. It's a typing error, please. It's A. It's A. Your answer is what? Blood therapeutic what? Agar or Lufla or Lufla or Lufla. So please, this is it. And it's easy to remember some of what? These things. Very easy to remember them. Very easy to remember them. So your answer is what? It's A, not D. Please, it's A, not D. Even this I don't know what it means. Your answer is A, please. It's A. Now, in Salmonella and in E. coli, you could think of what? Of endoagar, right? Exactly. You could be thinking of what? Endoagar. Great. So your answer is A. Ladies and gentlemen.
Please, you said Salmonella and what?
Salmonella and E. coli. Escherichia coli. That's Salmonella and E. coli. And I said that how do we differentiate between them? Is that this E. coli will produce a color or is colorful, while Salmonella is colorless. Salmonella is colorless. Thank you.